Hello friends, good afternoon and welcome to EduSite Live Lectures. Dear friends, today in genetics we will be talking about genetic recombination in bacteria transformation. To discuss this topic we have with us our subject expert Dr. Virendra Singh Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Hindu College, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome Sir.
Thank you. Friends, today I will be talking about a very important aspect of genetic recombination in bacteria, that is transformation. To underline the word recombination, what does recombination mean? As the name suggests, recombination in organism is basically recombining the genetic information. And when we talk about bacteria, it is basically adding up of information from some other species of bacteria in a host organism or a given organism. Because as we know that prokaryotes or bacteria do not reproduce sexually. Recombination is a very important feature in the genetics of bacteria because it leads to evolution of new strains, and because of recombination new characters, new genes are taken up by bacteria, and this imparts a new type of phenotype on the given strain of bacteria. So it is a very important evolutionary event. So the outline for today's lecture would be, I will be talking about basically about genetic recombination, what exactly is recombination, and in detail I will be talking about transformation. What exactly is transformation, how does it go about, how is it carried out in bacterial species. So the learning objective for the today's lecture would be that after this lecture we should be able to tell about natural and artificial transformation, how natural transformation occurs in bacterial species which are present in the environment, and how artificially we can transform bacterial species in the laboratory. Because as we all know that this particular aspect of science, biotechnology, that is transformation, is like being done in a major way throughout the laboratories in the world to study the genes and their control. So I will also be talking about how transformation can be brought about in bacterial species in under laboratory conditions. So as the name suggests, recombination. What exactly recombination is? Recombination is the process by which pieces of DNA are broken and recombined to give rise to new combination. So this is basically a layman's definition of recombination. In eukaryotic cells, recombination occurs during meiosis when the homologous pairs of maternal and paternal chromosomes align. So in eukaryotes, recombination is basically an event which occurs in gonads when the germ cells they undergo meiosis and there is aligning of the paternal and maternal chromosome, and they exchange part of the chromatid. The crossing over between the chromosomes lead to exchange of genetic material between the maternal and paternal chromosome, and the offsprings which are formed after fusion of such gametes, they have a different combination of genes than their parents. So in both the male parent and the female parent, the homologous pair undergo recombination, and the sperm and ova from the father and mother respectively, when they fuse to form a zygote, a new combination of gene arises because of this particular event of crossing over. So this is how recombination occurs in eukaryotes. And recombination is basically responsible for creating genetic diversity at the levels of genes, and this is reflected in the DNA sequences of the organism as well as in the phenotype which is caused because of such new recombination. So the offsprings are very much different phenotypically from the parents because of this event of homologous recombination in eukaryotes. But as we all know that prokaryotes do not have sexes, they do not have male parent or female parent, and they do not reproduce sexually. Though they do not reproduce sexually, still there are mechanisms or methods by which there can occur recombination in prokaryotic species. So So it is very well seen in many species of bacteria and characterized in many species like E. coli that recombination does occur. There is exchange of genetic information between the members of a species and sometimes between various species of the same genera. And it can also occur that there is genetic information exchange between species of different genera also. So there are basically three mechanisms which are responsible for bringing about recombination in genetic information of the prokaryotes, and these are namely conjugation, transformation and transduction. These are three different mechanisms through which recombination occurs. Now, what is difference between the three mechanisms, how they are different from each other? Though they are bringing about the process of recombining the genetic information, still the mechanism by which they occur or the mechanism which leads to recombination is different in all three of them. So, also because of different mechanism of occurrence, they also have different probability of occurrence. The probability of occurrence of the three gene transfer mechanism is because of specific requirements of the three mechanisms. So they all have different probability of occurrence in nature. The greatest requirement is that of conjugation. In this case, the donor cell must contain a conjugated plasmid, and there must be established a stable physical contact between the donor and recipient cell so as to allow transfer of DNA. Also, it is essential that both the cells be metabolically active to allow DNA synthesis and other activities. So, as these lines tell us that conjugation is the one which requires most of the input from the bacterial species. The most important input is that the species which are going to conjugate, one of the strain must not be having a conjugated plasmid, whereas the other strain which is going to transfer its DNA into the host organism, it should be having a conjugated plasmid. Unless there is a conjugative plasmid present in the bacterial species, it cannot initiate conjugation. So the presence of conjugative plasmid infers on the bacteria a condition which leads to formation of a conjugation tube and transfer of genetic material, that is DNA, into the host species. Also, the second important thing is that there should be a physical contact. The conjugation tube, which is formed because of presence of specific genes on the conjugative plasmid, this leads to physical contact between the donor and the host. So the genes are responsible for formation of tube. Once this physical contact is established, then there is transformation of genetic information from the host to the donor. Also, the cells should be physiologically or metabolically active so that there can be DNA synthesis in the donor organism as well as the host organism which is receiving the DNA from the host organism. So this particular process requires a lot of input from the bacterial species. Therefore, its probability of occurrence is quite low. Whereas in case of transduction, there is requirement of a metabolically active donor cell in which transducing phage particles are produced during viral reproduction. So apart from requiring a metabolically active cell which is carrying out metabolic process very actively, it also requires infection by phage particles. Either they are infecting newly or they are present in the chromosome of the bacteria. It does not entail any cost to the gene of the organism, that is, no genetic information of the bacteria itself is required for carrying out genetic recombination. Instead, it requires that cell be metabolically active and it should be having phage particles which are capable of transduction, because transduction is the process which brings about genetic recombination through infection of new species. So the transducing phage particle, it packs the DNA of the host organism, and when next it infects the other bacterial strain, it will transfer that piece of DNA into the donor into the host organism, and this will bring about genetic recombination. So there also is a certain cost to the organism, or certain requirement from the organism for carrying out this process. The recipient can be specially and temporarily separated from the donor in the case of transduction, because the genetic information in the transducing particle can persist. Whereas in the case of conjugation, there has to be physical contact between the donor and the host. In the case of transduction, there's no requirement of physical contact between the donor and the host. Also, they can be spaced out in time. That means it is not necessary that the host and donor are present together at the same time, because the bacteriophage will be packing the DNA of the host, and it can infect the donor, and it can infect the host after some time. So there can be there can be a time gap and space gap between the host and the donor organism. However, the recipient must be related to the donor by common sensitivity to the bacteriophage, but extensive metabolic activity is not required for the infection process. One important requirement is that the bacteriophage which is infecting a given donor species, it should also be able to infect the other species to which it will be transferring the genetic material. If the bacteriophage is not able to infect the host organism, then it won't be able to transfer the genetic information in the form of DNA. So it is very important that this particular bacteriophage can infect both the host as well as donor organism. Now the third process, which is known as transformation, it does not even require a living cell. So this underlines the entire difference between the conjugation and transduction and transformation, because the process of transformation just requires presence of DNA. It does not require formation of conjugation tube. It does not require physical contact between the donor and the host organism. It does not require infection by a bacteriophage. It just requires presence of naked DNA in the surrounding for the host organism to pick up that particular genetic information. And this can happen whenever there is lysis of the bacterial cell. It can release DNA into the surrounding, and this free DNA can be easily picked up by the bacteria present in the medium or the surrounding. So the persistence and dissemination of DNA in the environment determine how far in time and space the recipient cell can be separated from the donor. The only caveat or the only drawback for this particular mechanism is that it depends on free DNA, and free DNA, as we all know, that it is subjected to digestion by nucleases, which are very much prevalently present in the environment. So the only drawback is that this process can occur as long as DNA is not damaged or DNA is not cleaved by nucleases. As long as DNA is not cleaved, it can help in transformation of the host organism. But if the DNA gets cleaved by nucleases released in the environment, then there cannot be any transformation. Again, the recipient must be physiologically active to be able to take up DNA. If the recipient is not physiologically active, if the genes are not getting expressed properly, then this particular host organism will not be able to take DNA from the surrounding. So as we see that for all the three mechanisms, that is conjugation and transduction and transformation, host should be physiologically active. It must not be in a quiescent stage. If it is so, then these processes might not take place. And also, a close genetic relationship between donor and recipient cell is not necessary for transformation with DNA. As we saw that in conjugation, the strains which are bringing about conjugation, wherein the conjugation is occurring, they must be related, they must be different strains of the same species. Whereas in the case of transduction, there must be some relatedness in the fact that bacteriophage must be able to infect both the strains of the bacteria. Whereas in the case of transformation, there is not required any specific relationship between the donor and the host bacteria, because the free DNA is encountered by any cell, and if it is competent, if it is able to take DNA from the medium, then it gets transformed. So there is no requirement for genetic relationship between the donor and the recipient cells. Now, naturally, genetic transformation in bacteria occurs when there is active uptake by a cell of free DNA, that is extracellular DNA, which is not present inside the bacterial cell, and this DNA could be plasmid DNA or chromosomal DNA, and the heritable incorporation of its genetic information. Apart from uptaking of the free DNA, it is imperative, it is necessary that this DNA be incorporated in the chromosome of the host organism. If it does not get incorporated, then this piece of DNA will be sooner or later it will get degraded by the cellular enzymes, and the progeny of this particular cell will not be transformed cell. They will continue to be as they were. That means their genetic component will be as it was before uptaking of the DNA, and there will also not be any phenotypic changes in this particular host organism. This transformation again is as we all know, all these methods are a mechanism of horizontal gene transfer, and it depends on the function of several genes located on the bacterial chromosome. So, transformation is a mechanism of horizontal gene transfer where there is transfer of information from one species to another species, and it is dependent on expression of several genes which are present on the bacterial chromosome. A transformation can be considered the genuine bacterial gene transfer process, since other gene transfer processes are determined by genes located on plasmids and transposons like conjugation, and on bacteriophages, that is transduction. So, the other processes are requiring presence of certain genes. Like in conjugation, specific genes are required which form conjugation tube. Also in bacteriophage infection, that is transduction, again specific genes are required, but in the case of transformation it does not require presence of any extra genes. This process can be carried on by itself, though it requires that the cell which is uptaking DNA it be a competent cell. That means it is expressing certain competent genes. The bacteria are the only organism which are capable of natural transformation. Several independent lines of research have provided convincing evidence that transformation occurs in the environment, and the term natural genetic transformation has been coined to distinguish it from other in vitro or artificial procedures which are used to introduce DNA molecules into bacterial cells. Natural transformation of bacteria was detected more than about 5 decades ago in laboratory experiments by Frederick Griffith in the year 1928, when he injected mouse with a strain of a bacteria, and he found out that this particular bacteria was able to kill the mouse if it was a smooth strain, if it had a smooth capsule of polysaccharide. Whereas if the rough strain was injected into the mouse, then the mouse was not getting killed. When he took the smooth strain bacteria and he killed them with heat, and he injected this along with the rough strain bacteria and injected that into the mouse mouse got killed. So this occurred because there was uptake of DNA by the rough strain bacteria from the smooth strain bacteria. The DNA of the smooth strain was taken up by the rough strain bacteria, and this led to killing of the mouse, and this was the first documented evidence of transformation. Now there are many methods by which artificial transformation can be done, and these include treatment of cells with solutions of calcium chloride or chlorides of other elements, treatments of cells with chelating agents like EDTA, treatment of cells with enzymes leading to the formation of spheroplast or protoplast, fusion of cells or protoplast with DNA, with cells or with DNA packaged in liposomes, usually with the help of polyethylene glycol, or by freezing and thawing of cells, or exposure of cells to electric fields, that is electroporation. This is how transformation is carried out in laboratory conditions. Also bombardment of cells with small particles, thus transporting the DNA into the cytoplasm, can also be done in the laboratories. Now the bacteria which are present in the environment, they may encounter similar conditions. For example, a decrease in temperature below the freezing point of water, presence of solution of certain electrolytes, and the presence of lysozymes and proteolytic enzymes. These are the situations which are more or less likely to be found in aquatic and terrestrial environments or within higher organisms. So the conditions similar to laboratory can be created in the environment, and this can lead to natural uptake of DNA by the bacterial species, leading to transformation. High concentrations of protoplast cells are present in certain habitats like certain habitats, and certain species like Mycoplasma species and Rhizobium species can be found in the nodules of leguminous plant roots, and herein specific transformation can take place. This suggests that spontaneous DNA transfer can occur in natural bacterial communities. However, natural transformation depends on a set of functions which are carried out by the product of genes dispersed over the chromosome, and this is done under the influence of particular environmental conditions. This suggests that cells actively respond to conditions in the habitat by coordinating their level of gene acquisition through natural transformation, rather than being passively subjected to environmentally enforced gene exchange. So there is an interplay of the environment and the genes which are responsible for carrying out transformation. Taking the cue from the environment, these genes can get expressed, and in turn they lead to uptake of DNA which might be present in the surrounding. So there is an interplay going on between the expression of genes responsible for transformation and the environmental conditions. Now there are several stages of transformation, such as binding of DNA, its processing and transport into the cytoplasm, and the processes necessary for the propagation of the DNA. So these are the steps. That is, first DNA will bind to the organism, it will be uptaken into the organism, and then after processing by certain enzymes, this DNA will get integrated into the host chromosome. There are two main DNA uptake routes which have been described, and these are basically Streptococcus, which is a gram-positive bacteria, and the Haemophilus, which is a gram-negative bacteria. These are the two mechanisms of transformation. In these way transformation can occur. From the recent examination of species capable of transformation, it appears that some of the bacteria share features of both the models, that is Streptococcus and Haemophilus models. In Bacillus subtilis and Staphylococcus pneumoniae, double-stranded DNA associates rapidly with competent cells to form a complex that is resistant to gentle washing or to replacement by excess DNA, but it is sensitive to DNase treatment. At acidic pH, Bacillus subtilis also binds and takes up single-stranded DNA. About 50 and 30 to 80 DNA binding sites per competent cells are present in Bacillus subtilis and Staphylococcus pneumoniae respectively, and these seem to be involved in non-covalent association. Haemophilus influenzae, on the other hand, can bind and take up double-stranded and single-stranded DNA, but it can take single-stranded DNA only at a low pH of 4.4, which is an acidic pH. It binds and takes up double-stranded DNA only from the same or closely related species. So there is a limitation in Haemophilus influenzae system that it can take up DNA only from the genetically related species. This is because of a presence of a specific 10 or 11 nucleotide 10 or 11 nucleotide sequence, which is specific to this particular species of bacteria, and this is recognized by the competence factor which are present on the cell wall of this particular organism. In addition to the presence of a recognition site, that is 11 nucleotide sequence, the adenine and thymine, that is A and T content of the flanking DNA region also influences the binding and uptake of a DNA molecule in Haemophilus influenzae. At low pH, heterologous DNA can be bound. It suggests that non-specific uptake also occurs. When we lower the pH, then this organism is able to take DNA which is not related to this particular species. It can take DNA from other non-related species also. About 4 to 8 sites for DNA binding and uptake are present per cell in Haemophilus influenzae. Shortly after binding to the cells, S. pneumoniae cells, single strand breaks about 6 kb apart. These are introduced into the DNA by an endonuclease activity of the cell surface DNA receptor. This endonuclease is present on the surface of the cell, and uptake is defined as a transition of bound DNA into a DNase resistant state. It is not yet clear whether this occurs by movement of DNA into a vesicle into the periplasmic space or directly into the cytoplasm. The mechanism is not clear as yet. The transition is initiated by the generation of double-stranded breaks through cleavage at an opposite site near the initially introduced nick. The fragmented double-stranded DNA associated with the cell and still in a DNA-sensitive state participates in the entry into the cell, and after uptake, DNA is recovered from inside the cell as single-stranded material, and this particular uptake requires presence of calcium ions. And recent investigations have shown that the entry is polar, which starts at the 3' end of the nicks introduced during binding. Concomitant with the entry, an amount of labeled acid-soluble material equivalent to that of acid-precipitable material taken up is released into the medium, which indicates degradation of the complementary strand to the entering strand. And during translocation into the cytoplasm, the single strand becomes complexed with a protein which has a high molecular weight. One of the functions of this protein may be the protection of the single strand against nucleases. Another function may be to facilitate recombination. The processes that lead to uptake of bound DNA in Bacillus subtilis parallel those in S. pneumoniae, and the use of uncoupling agent has revealed that uptake is an energy dependent process which is driven by electrochemical gradient. Several models have been proposed to explain the energetics. One model, though not explaining the requirement of a nucleus, proposes that DNA passes through aqueous channel into the cell. However, there is a marked difference between S. pneumoniae and H. haemophilus system regarding the steps following the first reversible association of DNA with cell surface structure. Only specific homologous DNA is bound by Haemophilus influenzae under normal conditions. Subsequently, DNA is rapidly turned into a DNA resistant stage without apparent production of equivalent amount of acid-soluble material as described for the gram-positive species. During competence development, membrane vesicles with DNA binding capacity are formed in Haemophilus influenzae, and these vesicles are known as transformosomes, which extend about 35 nanometer from the cells, are 20 nanometer in diameter and are located on the cell surface where the inner and outer membrane appear to be fused. Homologous DNA is taken up as a double strand in an unknown way into the transformosome where the DNA is protected against exogenous DNAs. After a lag of less than 5 minutes, linear DNA is transported into the cytoplasm. From where does this DNA comes? It is released into the surrounding whenever there is lysis of the bacterial cell, and this leads to uptake of DNA by the host cell, which is present in the similar medium. So this is the basic process how transformation occurs under natural and artificial conditions. Thank you. Friends, I will be talking about genetic recombination in bacteria and how it occurs, the molecular mechanism behind this particular genetic recombination in bacteria through transformation. Now, transformation is a process wherein the bacterial species or the bacterial cell, they uptake free DNA from the medium, and this particular DNA, fragment of DNA, it gets integrated into the host chromosome. After it gets integrated, there is basically acquisition of the genes which were carried in the free DNA, and also there is phenotypic change in that particular species and also in the progeny species. So what exactly is transformation? How is it brought about? What are the molecular factors or molecular mechanisms which are responsible for bringing about this transformation in the host species? So I will be talking about this particular aspect in today's lecture. So the outline for today's lecture, I will be discussing about competence in the bacterial cell, competence is basically competency or the ability of the bacterial cells to uptake DNA from the surrounding medium. This could be culture medium in laboratory, or it could be natural environmental medium wherever that bacterial species is thriving or growing, thriving and multiplying. Also, I will be discussing about how DNA is uptaken by the competent cells, what is the molecular mechanism behind that, and how there is integration of DNA in the host chromosome after the DNA is uptaken by the cell. And then I will be discussing what is the outcome of transformation, how exactly the transformation changes the bacterial species, what changes it brings about, and how this particular mechanism of genetic recombination can be used for genetic mapping, how it can be used for assigning specific genes to specific regions of chromosome of the donor DNA or of the plasmid of the donor DNA. So, the learning objectives for this particular lecture would be that after this lecture we should be able to tell about the process of development of competence in the bacterial cell, that is how competence develops in the bacterial cell in the course of transformation, how the DNA is uptaken and integrated into the host chromosome, and how this particular mechanism can be used for genetic mapping of genes in the donor organism. So, what is competence? What exactly this word means in the scientific language? The genetic competence is defined as the ability of a cell to take up free DNA from the surrounding medium. Any bacterial species which is able to take up free DNA is said to be a competent cell, and competence is defined as the ability, that particular ability of cell. Herein it is important to underline that not all bacterial species will be competent at all period of time. Suppose if we take a bacterial species in the environmental condition or in laboratory condition and we put some free DNA in the medium or in the environmental condition, it is not, the bacterial species is not predatory, it will not start taking up free DNA as such unless it develops competence, unless it has competence factors in its membrane, then it won't be able to uptake DNA. Also, bacterial species or the bacterial organisms are not competent at all phases of their life, at all point of their growth phase. They are competent only for a given period of time, a specific period of time, and in a specific phase of their life cycle. So, it is wrong to assume that if we provide the free DNA, bacteria will uptake the DNA. There is going to be a specific set of conditions which will induce competency in the bacterial cells, then only they can uptake DNA. In several instances during growth, under specific conditions, cells develop the capability to bind and take up DNA. As I told earlier, that there are specific conditions under which the bacterial cells will develop the ability or the capability to bind and uptake free DNA from the surrounding. Now, development of competence depends on the expression of the genes and the proteins encoded by which carry out the necessary function. Bacteria on its own as an organism, as a being, cannot uptake DNA by its own will unless the genes which are responsible for expression or forming of competence factor protein, unless they are getting expressed, the bacteria cannot uptake DNA. So, this particular property of competence, it is widely distributed among the taxonomic and trophic groups, which include archaebacteria also. So, this particular property of having competence, this suggests a long evolutionary history of development, which means that competence evolved a long way back, because it is also present in the most primitive of bacteria known as the archaebacteria, and it has continued unabated and into the new bacterial species which evolved from these organisms. So, it is a widespread phenomenon occurring in the prokaryotic world. Now, in most naturally transformable bacteria, competence is transient. Transient means it is a very small phase. So, all the bacteria which are occurring naturally and which are carrying out transformation naturally, this particular phase is very small lived phase. In Neisseria gonorrhoeae, however, the competence is constitutive. That means this bacteria is continuously competent. It can uptake DNA at all times. In other transformable bacteria, that is bacteria which can be transformed, which can uptake DNA from the surrounding. This particular property of having competence, it is an inducible physiological property. That means this particular physiological aspect can be induced by providing it certain specific environmental conditions. In Haemophilus influenzae, the development of competence starts when the cells are transferred to a media which does not allow growth, or when the cell division is blocked under conditions permissive for protein synthesis. So, as we see here, the competence development is starting whenever the cells are transferred to a media which is not allowing the cell to grow, or when the cell division is blocked. Now, competence usually arises at a specific stage of growth of a culture, and this phase is usually the late log phase and highly depends on the growth medium and the degree of aeration of the culture. As I told already that competence is not present, competency is not present in the bacterial cell throughout its cycle. It is present at a specific stage, and it usually arises at a specific stage, and in most of the bacterial species this phase is the late log phase. And it also depends on the nutrient medium, which is present in the culture medium, and how much the culture is aerated. The particular medium and growth conditions that produce maximum competence vary from species to species. So, it means that the competence of different bacteria, it varies according to a specific condition. If we provide a specific set of conditions to all the bacterial species, it does not mean that all the bacterial species will develop competence. For each species, there has to be a specific set of environmental condition and growth condition, culture condition, which will induce competency in that particular species. That particular set of conditions will not work for another species. For the another species, another set of conditions will have to be given to induce it into competency. Now, competence develops as cells begin to grow and reaches its maximum during the early to late log phase in Acinetobacter, Azotobacter, Staphylococcus aureus, Streptococcus pneumoniae. So, in these particular species, competence starts developing as the cells start growing, and it is maximum in the late log phase of the cycle. However, in Bacillus subtilis, it occurs during transition from the log phase to stationary phase. This underlines the fact that for each species of bacteria, there has to be a specific set of conditions, and competence will develop in its own specific stage. There is no set rule that competence will develop at a specific stage of the life cycle. In Mycobacterium, competence proceeds throughout exponential growth and decline during the stationary phase. So, I have given three types of example. In late log phase, there is Streptococcus pneumoniae in Bacillus subtilis. It occurs during transition from log phase to stationary phase, and in Mycobacterium, it starts throughout the exponential growth, but it declines during the stationary phase. So, this underlines the fact that for each species, there has to be a specific set of condition, and competence occurs at a specific stage of the cycle. Of the culture. Now, the competent fraction of a population may make up only a small percentage of the culture, as in Streptomyces virginiae, or between 10 to 25% as in Bacillus subtilis, or almost 100% as in Haemophilus influenzae and Streptococcus pneumoniae. So, basically, how many cells at a given time in a culture medium are competent, that again varies from species to species. If we say that culture has developed competence, it does not mean that all the cells in the given time in the culture medium are competent. That means they are able to uptake the free DNA. For each of the species, there is a specific set of conditions, as I have already told, and the percentage of cells which develop competence is varying from one species to another. In case of Streptomyces, the percentage is very minimal. It is about 10 to 25% in Bacillus subtilis, whereas all the bacterial cells present in culture medium of Haemophilus influenzae and Streptococcus pneumoniae, all of them will be competent at a given stage of time of the life cycle. However, in Streptococcus pneumoniae, competence persists for only a few minutes, whereas in Bacillus subtilis, the competent state lasts for several hours. So again, there is a difference in two species, whereas in one species, though all the cells are competent, that is, there is 100% competency, in only if this particular stage lasts only a few minutes. Whereas in another species again, which has 100% competency, the competent state lasts for several hours. Many species have been studied, and this particular aspect varies from one species to another species. And also the species which have not been able to, which the scientists have not been able to study for transformation, in them also we assume that there is a different set of condition for inducing competency, and we have not been able to pinpoint what specific set of conditions will induce those cells to to start making competence factors and undergo transformation. At a critical cell concentration, competence development in S. pneumoniae and B. subtilis, that is Bacillus subtilis, is induced by an excreted small polypeptide called competence factor. That is, whenever the cells are being cultured, the cells increase in number, first at a slow pace, then exponentially, and then the number starts declining. The increase in number starts declining at the plateau phase. So at a specific concentration of a cell, there occurs development of competence in S. pneumoniae and B. subtilis, and this occurs because a small number of cells, they start expressing the gene for competence factor, and this particular gene, when expressed, it forms the competence factor protein. This particular protein gets excreted or get excreted into the surrounding medium, and once this protein is in the medium, it will be inducing the related or the other cells present in the medium to undergo competency, to start making competence factor. Competence appears to be a result from the changes in the cell wall of the bacteria, and is probably associated with the synthesis of cell wall material at a particular stage in the life cycle of bacteria. So basically, it is assumed that whenever there is change in the cell wall of the bacteria because of a specific condition, specific culture condition, and also because of it being in a specific stage of the growth of that particular medium, this leads to synthesis of competence factors. Now, for developing competence, receptors of some type are either formed or activated in the cell wall. It is suggested that receptors, which are again proteins, receptors of a specific type are required to be formed either on the cell wall, or they are present beforehand, and because of the specific conditions which are attained, these receptors are activated, and they lead to activation of the cell for taking up DNA. Now, these receptors are responsible for initial binding of DNA. The receptors which are present on the cell wall of the organism, these are responsible for binding of free DNA which is present in the medium. The number of active receptor varies widely from one organism to the organism. There is a great variety in the number of receptors which are present on the cell wall from one species to another species. There is no fixed number for any of the species. There is a great variation in such receptors which are present in the cell wall of the organism. But it is to be underlined that competence initially arises in a small fraction of cells, and the cells which become competent, they secrete competence factors into the media, and they convert the remainder of these cells into competent state. So it must not be assumed that at a specific stage of the growth of the cell culture, all the cells simultaneously become competent. Initially, only a few of the cells, they become competent, and because of these competent cells, they secrete competence factors into the medium. These competence factors, they cause the remainder of the cells to become competent. The remainder of the cells might be about 10 to 25% as in Bacillus subtilis, or all of the remainder cells which are present in the medium as in S. pneumoniae culture. So this varies again from one species to another species. Now, it has been seen and it has been observed in experiments that purified competence factors can also induce competency in a bacterial cell culture, irrespective of the growth phase of culture. As I have told earlier that the development of competency is dependent on the growth phase of the culture, in which particular growth phase that particular culture is, and it is varying from one species to another species. But if the competence factors, which are proteins, if they are isolated and purified, and if they are put in the culture medium, then it will lead to formation of competent cell cultures, irrespective of whichever growth phase that particular culture is in. It can be in the lag phase, it can be in the exponential phase, it can be in the log phase. This suggests that the medium, degree of aeration, the temperature needed to produce a competent culture serve only as a trigger to synthesize or release competence factors by a small number of cells. So, all the environmental conditions which I talked about, they are required just for synthesizing or releasing of competence factors by a small number of cells, and in that way they act as a trigger. After a small number of cells start synthesizing these factors, then the remainder of cells, which again is varying from species to species, they also become competent. The competence factors are species specific and induce competence only in closely related species. If we take purified competence factors from species A and we try to induce competency in species B using that particular competence factor, it will not be able to induce competency because they are species specific. Also, if we culture two different bacteria simultaneously in a culture medium, and of one of the species, that is species A, few cells develop competency and they secrete their competence factor in the medium. The species B, they will not be turned into competent cells. Only the species A will turn into competent cells because of competence factors limitation, that they are species specific and they can induce competence only in species which are closely related. Now, once the cells are competent, they can uptake DNA. This is the basic requirement or the primary requirement for transformation, that cells develop competency. Once they develop competency, only then they can uptake DNA. Now, DNA uptake by competent cells has been examined in two ways. In one type of experiment, cells are exposed to radioactive DNA and then to a DNase which is capable of degrading DNA completely. Now, experiment suggests that there are at least two stages of interaction of DNA with competent cells. So, what we are doing is we are exposing the cells, bacterial cells, to radioactive DNA, and after some time we are exposing these cells to DNases which are able to denature or degrade the free DNA completely. Now, the experiments have suggested that there are two stages in the interaction of DNA with the competent cells. The first stage is a brief reversible binding stage in which DNA can be washed from the cells and in which DNA is completely sensitive to DNase degradation. That is, the DNA is bound to the cell, but it can be digested by DNase. And then there is the second stage or irreversible stage, wherein DNA cannot be washed from the cells and is resistant to DNase. So, in this particular stage, DNA is though bound to the cell, in this particular stage it cannot be cleaved by DNase. Irreversible interaction is an energy requiring process that is associated with passage of DNA through the cell membrane. Whereas reversible first stage or reversible stage, DNA is just bound to the surface of the cell. In irreversible stage, it transfers from the outside of the cell into the inside of the cell through a channel, and it requires energy consumption. DNA uptake is not species specific because the DNA molecules are same for all species, though the nucleotide sequence might vary. Whereas the competence factor are species specific, that means they can induce competency only in the same species. Once the competency has been induced, the DNA which is present in the medium, it can be uptaken irrespective of it is coming from the same species or it is coming from some other species, because DNA is just a sequence of deoxyribonucleotides, and this particular sequence is not recognized by the competence factors. They recognize only the sequence of nucleotides, and it can be uptaken irrespective of the species. However, There are certain species which will uptake only specific DNA, like Haemophilus influenzae, which takes up only DNA of its own species, because it has the ability, the competence factors have the ability to recognize sequences, nucleotide sequences of 10 base pair, which are needed for initial binding of the DNA to the cell surface. So, this particular species is specific for DNA uptake, while others are, most of the species are not specific for DNA which they are uptaking. Now, a competition assay is used to show that a particular DNA molecule can be taken by a competent cell. Now, what is this assay? In one experiment, different samples of penicillin sensitive S. pneumoniae were mixed with a fixed amount of DNA isolated from penicillin resistant culture and with various amount of calf thymus DNA. That means we have two types of DNA, calf thymus DNA and DNA isolated from S. pneumoniae, which is resistant to penicillin antibiotic. Increasing the amount of calf thymus DNA reduced the number of penicillin resistant transformants. The foreign DNA, that is calf thymus DNA, binds to the receptor sites and therefore blocks the uptake of the S. pneumoniae resistant DNA by S. pneumoniae sensitive DNA. So, because of presence of a competitor which is able to bind to the competent site, the frequency of transformation of sensitive cells is decreased appreciably. So, this suggests that the calf thymus DNA, which is a eukaryotic DNA, it can also be uptaken by the bacterial cells. Now, denaturation of DNA sample capable of transformation, it eliminates all the transforming activity. That means DNA is required to be present in double stranded form. If it is present in single stranded form, if we denature the DNA into single strand, it won't be uptaken by the bacterial cell. Addition of competing but denatured calf thymus DNA does not inhibit transformation. This suggests that even the calf thymus DNA, which is single stranded, it is not binding to the receptors present on the bacterial cell. However, if the denatured DNA is renatured, then both the transforming activity and the ability to compete with normal transforming DNA are restored. So, it underlines the fact that the DNA has to be present in double stranded form. Single stranded or denatured DNA cannot be uptaken by the bacterial species. Now, the state of DNA when it is uptaken by the cell, it gets altered during uptake. A streptomycin sensitive culture of S. pneumoniae is in an experiment is exposed to DNA from the streptomycin resistant strain. After 2 minutes, cells are washed free of excess unincorporated DNA and divided into two portions. The first, after a sufficient period of time to allow expression of the streptomycin resistant gene, one part is plated on a medium containing streptomycin and streptomycin resistant colonies were obtained. The other part is used to isolate DNA, but when it is used to transform the sensitive S. pneumoniae culture, it does not show any transforming activity. Whereas, if the same experiment is repeated at 15 minutes, similar set of conditions, in the first part similar number of resistant colonies were obtained, but in the second part, when DNA is isolated from such cells, it is able to carry out transformation of fresh culture of streptomycin sensitive strain. What does it tell? It suggests that after adsorption and entry of DNA into the competent cells, initially it is non-transforming state and afterwards it is converted into a transforming state. The period during which no transforming DNA can be isolated from the potentially transformed cells is called eclipse. So, this is the period when no transforming DNA can be isolated from such cells. Now, how this occurs? There are two possible mechanisms that this occurs, that there might be two possible mechanisms which lead to transformation. One is that incoming donor allele replaces the allele of the recipient, that is the DNA fragment containing the donor allele is added to the genome of the recipient by insertion adjacent to the recipient allele, thereby forming a partial diploid organism. By study of reciprocal transformation, it was confirmed that there was no addition of allele during transformation. The second experiment, which used two closely linked markers, confirmed that there is no addition of alleles. If there is a recipient bacteria which has two closely linked alleles, say small m and small n for antibiotic sensitivity, and the donor was resistant, it was capital M and capital N, then three possible genotypes can be formed as shown in the slide, and these were found with equal frequency, which shows that M and N genes are linked. In a second experiment, capital M and small n was used to transform a small m and capital N recipient, and M marker was selected as a phenotype. If addition occurred, then genotype would be either capital M, small m and capital N, and capital M, small m, capital N and small n, both of which would be displaying N phenotype. But it was seen that colonies of both capital M and capital N and capital M and small n phenotypes were present. This suggests that there is allelic substitution, there is no insertion or partial diploid condition during transformation. So, in this particular case, there is incorporation of M. It is often associated with removal of N marker of the recipient and replacement by small n, which was carried on the same transforming DNA molecule as the M marker. Integration into the recipient chromosome seems to be similar in Streptococcus and Haemophilus types of transformation. After the single-stranded chromosomal donor molecules have entered the cytoplasm, they are integrated in homologous regions of the recipient chromosome. And the heteroduplex is thought to be formed by assimilation of the donor strand concomitant with the displacement of the corresponding recipient strand. In Bacillus subtilis, about 70% of the single-stranded homologous DNA taken into the cytoplasm is integrated. Now, the formation of donor-recipient complex is catalyzed by REC-A-like protein, and the role of this protein in heteroduplex formation is substantiated by a number of experiments. And the mutants which are deficient in this particular protein, they cannot undergo transformation. And this particular protein causes recombination even between non-matching nucleotide pairs. So, this particular figure shows what exactly happens during transformation. The double-stranded DNA binds to the plasma membrane, and this particular double-stranded membrane double-stranded DNA is cleaved. One of the strand is cleaved, and the other complementary strand is uptaken into the cell through the receptor, and through this particular strand is then recombined with the chromosome of the host by using machinery which helps in cutting the host chromosome and insertion of this particular single-stranded DNA. After that, there is function of DNA polymerase which fills in the gap, and the ends which are formed after this particular incorporation, they are sealed by ligases. So, only a single strand enters the cell, and this particular strand is uptaken into the host chromosome, and the progeny will be having one normal host chromosome and one transformed chromosome. So, this is how transformation occurs in bacteria and brings about genetic recombination. Thank you.
On that note, I would like to thank sir for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.